Okay, so we're moving on with erythropoiesis. So hematopoiesis is the general term for the formation of the cells of the blood. If we're talking specifically about the red blood cells or the erythrocytes, we're going to be talking specifically about erythropoiesis. So erythropoiesis is the production of the erythrocytes, and this is going to occur in the bone marrow. Most of this blood cell production is going to occur in the bone marrow. So we already have talked about the hormone that is going to trigger erythropoiesis, and it is erythropoietin. We already talked about how it was produced in the kidneys, and it's going to be produced in response to hypoxia. Hypoxia is a decrease in the oxygen content of the blood. So when there is a decrease in the oxygen content of the blood, it triggers the hormone or endocrine cells within the kidney to release erythropoietin to raise red blood cell levels in an attempt to try to raise the oxygen content of the blood itself. The process through which erythropoiesis occurs is just like all other of the blood cell formations. We're going to start again with that hemopoietic stem cell. The hemopoietic stem cell is then going to become a myeloid stem cell. And the myeloid stem cell is then going to be acted on by some chemicals, the hormone erythropoietin, and that myeloid stem cell is then going to become the committed cell called the proerythroblast. And the reason why the proerythroblast of this committed cell is important is because once that myeloid stem cell has become a proerythroblast, there's no turning back. It is going to become a red blood cell. It can't go back to a myeloid cell and that myeloid stem cell and then become, let's say, a basophil or a eosinophil. It is committed. It's got the ring on the finger. It is going to be an erythrocyte. From an erythrocyte, proerythroblast, we then are going to go to another intermediate called erythroblast, which we're not really worried about. What I'm worried about is this guy right here in the middle called the reticulocyte. The reticulocyte is then going to mature into what we call the mature erythrocyte. So an interesting thing about the erythrocytes or red blood cells is that they do not contain nuclei. They also do not contain many they don't contain like organelles. So the erythroblast, as you can see here, has a nucleus. The proerythroblast has a nucleus. The reticulocyte, when it is first formed, has a nucleus, but before it enters into circulation, it's going to shed its nucleus. The reticulocyte enters the bloodstream, and after 24 hours in circulation, the reticulocyte then matures into the erythrocyte. Okay, so hemopoietic stem cell, myeloid stem cell, proerythroblast, committed cell, reticulocyte, erythrocyte. Thrombosis. So thrombosis is going to be, thrombopoiesis. Thrombopoiesis is going to be the technical term for the formation of platelets. Again, this is going to happen in the bone marrow. Anything that is T-H-R-O-M-B, is going to relate to some sort of clotting of the blood. So a thrombocyte is the technical term for a platelet. When we talk about a thrombosis, that is the formation of a blood clot. There's another, a thrombus is an actual blood clot. So when you see T-H-R-O-M-B, start thinking of something that does clotting, or it's going to be something involved in blood clotting. So the hormone that's going to trigger thrombopoiesis is thrombopoietin. See a little bit of a pattern here. Erythropoiesis, erythrocyte, erythropoietin, thrombopoiesis, thrombocyte thrombopoietin. Okay, so thrombopoietin is going to be a hormone that triggers thrombopoiesis. Again, this is going to be produced in the liver and the kidneys. If we look at the chain of events here, we're going to start with that myeloid stem cell. We can actually go out one more. That would be the hemopoietic stem cell. The hemopoietic stem cell is going to become the myeloid stem cell. Thrombopoietin is going to interact with the myeloid stem cell, and it's going to cause it to become the megakaryoblast, which is probably one of my favorite names for a cell. It sounds like a evil megakaryocyte. A megakaryoblast and megakaryocyte kind of sounds like a evil, villain in some Marvel movie or something. So the megakaryoblast is going to be the committed cell. Once that myeloid stem cell becomes the megakaryoblast, it is destined to become a platelet. The megakaryoblast is then going to become the megakaryocyte. And what happens to the megakaryocyte is that the megakaryocyte is going to undergo numerous rounds of mitosis, cellular division. And it's going to undergo this mitosis, but it's never going to undergo cytokinesis, which is the division of the cytoplasm. So the megakaryocyte is just going to get larger, 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 and larger, hence the name mega, because it's so large. And eventually what's going to happen is that that megakaryocyte is going to start to form these little tiny cytoplasmic extensions off the edge of it, because it's getting so large. And it's these little tiny extensions are actually going to break off, and the cytoplasmic extensions that break off actually become the platelets. So realistically, even though we refer to the platelets or the thrombocytes as formed elements or cells, they're not really full cells. They're just little cytoplasmic fragments that have broken off a cell. Okay. Thrombopoiesis, formation of thrombocytes, aka platelets. We have the hemopoietic stem cell to the myeloid stem cell. Myeloid stem cell gets affected by the thrombopoietin, the hormone that is going to trigger thrombopoiesis. The myeloid stem cell becomes the committed cell megakaryoblast. Megakaryoblast becomes the megakaryocyte. Megakaryocyte undergoes mitosis, becomes larger and larger, larger, larger. We get these little cytoplasmic extensions. Portions of these cytoplasmic extensions break off. Those portions that break off are going to be our platelets or our thrombocytes. Leukopoiesis. So anything that is in Leuko, that is the Latin form, comes from the Latin word for white. So leukopoiesis or leukocytes, if we know cyte means cell, leuko means white. So these are going to be our white blood cells. Leukopoiesis is the production of leukocytes. Now this one is a little bit different because so far with the erythrocytes or red blood cells, we had one cell, produced from a line. In the thrombocytes, we had one, cell fragment produced from a line. Leukopoiesis gets a little bit more difficult because there's more than one type of white blood cell. The white blood cells that we have include the eosinophil, the basophil, neutrophil, listed here as a macrophage, but we have monocyte and then lymphocytes. So there's 5 different types of white blood cells. So it's going to be a little bit different because we have to talk about how each of the 5 different ones are going to be formed. So leukopoiesis, production of leukocytes, like the thrombopoiesis and the erythropoiesis, leukopoiesis is all of them, regardless of which of the 5 white blood cells we're going to talk about, is going to start off in the bone marrow. Most of them are also going to mature in the bone marrow, just like the thrombocytes did, just like the erythrocytes did. However, there's one leukocyte that does not mature inside of the bone marrow. It actually leaves the bone marrow, moves somewhere else, and undergoes maturation. And these are the T lymphocytes. T lymphocytes are going to mature outside the bone marrow, and they actually are going to mature in a organ called the thymus, and the thymus is actually located, it lays right on top of the heart. So let's look at how these guys are going to be undergo development. Well, we have 3 types of white blood cells that are grouped together in this group that's called granulocytes, and we'll talk more about that when we talk about white blood cells. And the way that they're going to be produced is that we have this hemopoietic stem cell. Here it's listed as pluripotent. That is a term that is used to describe a stem cell, but this is our hemopoietic stem cell. Well, actually it would be up here, hemopoietic stem cell. The hemopoietic stem cell is going to become a myeloid stem cell. That myeloid stem cell is then going to become a committed myelioblast. So myelioblast is the committed cell. So once it becomes a myelioblast, it's either going to then mature into a basophil, a neutrophil, or an eosinophil. Okay, so for these 3 white blood cells, the Phil family, I like to call them, the eosinophil, neutrophil, basophil, we have the hemopoietic stem cell, the myeloid stem cell, the committed myeloblast, and then that committed myeloblast is either going to become an eosinophil, a basophil, or a neutrophil. All right, that's the first 3. So let's talk about the 4th one. The 4th one is going to be the monocyte. Again, we're going to start off with that hemopoietic stem cell. The hemopoietic stem cell is going to form the myeloid stem cell. That myeloid stem cell is then going to become the committed cell, the monoblast. And then that monoblast is going to form the mature cell, the monocyte. Now, here in this picture, it's listed that the monocyte becomes a macrophage, and that's because when the monocyte is doing its work as a white blood cell, it actually becomes a macrophage, but it moves in circulation as a cell that's known as the monocyte. The last one we have is the one that doesn't really like to play by the rules. It's a little bit different, you know, a little special, that's how we say, and this is the lymphocyte. So again, though, it's going to start with that hemopoietic stem cell. Now, here's our first difference. The hemopoietic stem cell is going to become a lymphoid stem cell. Up until now, it's always been a myeloid stem cell. So the hemopoietic stem cell becomes a lymphoid stem cell. That lymphoid stem cell, for the case of the B lymphocyte, is going to become the lymphoblast, and I don't have it written here, I should. The lymphoblast is going to be the committed cell, and then that lymphoblast is going to become a B lymphocyte, which is also referred to as a B cell. This all happens in the bone marrow, this one does. So hemopoietic stem cell, lymphoid stem cell, lymphoblast, to the B lymphocyte, all in the bone marrow. And you can remember that it happens in the bone marrow because that's exactly why it has this B here, this B prefix before lymphocyte, to remind you that this happens in the bone marrow. Now, the other way that it can go is that this hemopoietic stem cell differentiates into the lymphoid stem cell. The lymphoid stem cell, again, turns into the lymphoblast. But at this term, the lymphoblast is going to become a T cell. That T cell leaves the bone marrow, moves to the thymus, and it's going to mature. And that's why it has the prefix of that T. The T stands for that thymus. So T lymphocyte is going to be the one who's going to jump ship, leave the bone marrow, move somewhere else, and finish its maturation process. All right, so now that we talked about how all of these guys are going to form individually, let's talk about their characteristics. So first one we're talking about is going to be the erythrocytes. So the erythrocytes we've already talked about, these are the red blood cells. Anything that is erythro is red. Good thing to remember if you ever see how to take like a standardized test. If you see erythro, you got to know it has something to do with inflammation, something that has to do with the red color. So the erythrocyte is going to be the most common formed element. Its primary function is going to be to transport gases. It's going to pick up oxygen in the lungs. It's going to transport that oxygen through the circulatory system and drop that oxygen off to the tissues, where the tissues are going to use it to undergo cellular respiration. Once it drops off that oxygen in the tissue, it's going to a limited degree, because most carbon dioxide is going to circulate dissolved in the plasma. It's going to pick up carbon dioxide that's in the tissues, carry that carbon dioxide back to the lungs, where it's going to be released, and we are going to exhale that carbon dioxide. I already mentioned this, but erythrocytes, mature erythrocytes, are going to lack nucleus and most of the organelles. Remember, when the cell is maturing, when it becomes that reticulocyte, the last thing it does before that reticulocyte moves into circulation for its final maturing or maturation process, it is going to eject or get rid of its nucleus. So the reticulocyte is an immature erythrocyte which has entered into a circulation. And here we have a staining of blood cells. And these blue cells right here are actually red blood cells. They are blue because they've been stained with a particular dye. The ones that have arrows pointing to them, these are reticulocytes. And what you can notice with them is that the reticulocytes have some little speckling in them. That little speckling is staining the remaining organelles that are still in the cell and are in the process of being degraded as that reticulocyte matures for that 24 hours before it becomes a mature erythrocyte. But as you can see, there is no nucleus present. The reticulocytes have ejected that nucleus before they leave the bone marrow and enter into circulation. Now, reticulates can be very helpful because if you take a blood sample and you count the number of red blood cells in circulation and compare that to the amount of reticulocytes in circulation, you can get an estimation of the rate of new red blood cell production. If you have a lot of reticulocytes in circulation, your body is in the process of producing a lot of new red blood cells. If there is a low reticulocyte count, then your red blood cell production has slowed down. If we look at the actual structure of a erythrocyte, it is what is referred to as a biconcave disc. So we normally think of a cell as being nice and round, nice, round, and plump. But what is actually making that cell nice, round, and plump is the nucleus that's on the inside. So if the nucleus is removed, what happens to the cell, it kind of like deflates in on itself, and it makes this biconcave disc. So this is it looking from the top view. If we look at the side view, we take a cut through it, you can see that both the top and the bottom portion kind of dips in, because the center portion used to be where the nucleus was taking up a lot of that area. Now, the biconcave disc is a very useful shape for the erythrocyte for two reasons. The first is that it actually provides a lot of surface to volume ratio. So there's a lot of surface area compared to the volume that is inside the cell. And this large amount of surface area is going to make it much easier for gases to enter and also to exit the red blood cell, because the gases are going to need to diffuse through the plasma membrane. So the more surface area they have to do that, the easier they're going to be able to. Also, red blood cells need to fit through very, very tiny capillaries. In fact, there's capillaries in the body where only one red blood cell is going to be able to pass through at a time. So this flattened disc-like shape is going to allow the red blood cell to kind of bend and move and squeeze through these very tight capillaries. In fact, inside of the erythrocytes, there is a cytoskeletal protein called spectrin, and this protein is very important because it's going to be able to allow the red blood cell to maintain its very unique shape, but also bend and fold and pop back into shape as it squeezes through these very tiny capillaries. Kind of like, there's pop-up tents where they kind of like fold up, and then, you can fit it in the bag, and then you pull it out and you go, and it goes poof, kind of think of spectrum as that. It allows the red blood cell to fold itself up when it needs to fit into a tight space, or for the tent to fit into a tight bag. But when it's moving through, when the red blood cell is moving through a larger vessel, that spectrin is going to allow it to pop right back up into this larger shape, just like those kind of a plastic poles allow those pop-up tents to just pop right back up into shape. Inside of the erythrocyte, there's a very important protein that we've mentioned before when we mentioned about the color of blood, called hemoglobin. And hemoglobin is often just abbreviated as capital H, lowercase b. So this is a large molecule that's made up of protein chains, polypeptide chains, and also heme and iron. There are 4 folded chains that make up this protein. So hemoglobin is what we refer to as a quaternary structure. If you remember back when we talked about protein structure, there was the primary structure, which was the sequence of amino acids, the secondary structure, which was either like a zigzag kind of folding or a spiral folding, alpha helixes or beta sheets or alpha helixes, sorry. And then we had a tertiary structure, which those beta sheets and alpha helixes kind of like folded in on each other and made a 3-dimensional shape. And then we talked about a quaternary structure where we had different polypeptide chains grouped together to make one massive molecule. So this hemoglobin molecule is this quaternary shape. And here we have 4 chains of proteins, and these proteins are globulins. We have 2 what are referred to as alpha chains and 2 beta chains. What is important about these 4 chains is that each of these globulin chains is going to bind to its own pigment molecule, and that pigment molecule that we talked about before is called a heme. So if each hemoglobin molecule has 4 polypeptide chains, then and each chain has a heme pigment, each hemoglobin molecule is going to have 4 hemes. Now, in the center of each of those 4 hemes, there is going to be an ion of iron. So there would be one here on this chain, one on this one on this one, and one on this one. All right, so quick review. 1 molecule of hemoglobin is made up of 4 polypeptide chains. Each one of those chains is going to contain 1 heme pigment. So if each chain has 1 heme pigment, that means each hemoglobin molecule is going to have 4 heme pigments. Each of those heme pigments is going to contain 1 ion of iron. So if there is 1 ion on each heme, and each polypeptide has 1 heme, that means each hemoglobin molecule has 4 polypeptide chains, 4 heme molecules, and 4 ions of iron. Now, why this is important is because each iron ion has the capacity or the ability to bind 1 oxygen molecule. And the reason why we're calling it an oxygen molecule and not an oxygen atom is because oxygen naturally forms a diatomical molecule, which means that it travels in pairs of 2. All right, so each iron atom can bind 1 oxygen molecule. And each hemoglobin transports, so that means that each hemoglobin transports 4 oxygen molecules. Why? Because there's 1 iron for 1 heme, and there's 1 heme for 1 polypeptide, and we know that each hemoglobin has 4 polypeptides. So that means 4 hemes, and that means 4 ions of iron, which means that's 4 oxygen molecules. Now, there's not just 1 hemoglobin molecule in a red blood cell. These erythrocytes or red blood cells are jam-packed packed with these molecules. In fact, each erythrocyte contains about 300 million hemoglobin molecules per red blood cell. So if we do the quick calculations, if one red blood cell has 300 million hemoglobin molecules, and we know that each hemoglobin molecule can carry 4 oxygen molecules, and we have this O2 here because oxygen molecule is made up of 2 oxygen atoms. That means that each red blood cell in your body has the capacity of carrying 1.2 billion molecules of oxygen. Just one red blood cell. And I don't even really want to, I can't even guess how many red blood cells you have in your body. I don't know what the number is. But if you think about the fact that, you know, the average adult has about 5 liters of blood in them, and 45% of that gallon is made up of purely red blood cells, and they're microscopic, and you can't see them with the naked eye. Just imagine how many red blood cells are in there, and each one has the capacity of carrying 1.2 billion molecules of oxygen. So red blood cells in your body have an amazing capacity for carrying oxygen. Now, when that hemoglobin is attached to oxygen or not attached to oxygen, it can be given a kind of special surname. So oxyhemoglobin is going to be when the iron ion in a hemoglobin is bound to oxygen. And again, because we said earlier that heme, that pigment, is going to change color based on the presence of oxygen or not, oxyhemoglobin is going to have that bright red color, that bright red lipstick color, or that bright Christmas red color. If there is no oxygen bound to that iron ion, we call it a deoxyhemoglobin, or D means without. So without oxygen hemoglobin. And in this case, the heme pigment has a more darker red color, more of a maroon color, that kind of like dark, vampy, red, brownish color that you think about. Now, I said that hemoglobin, red blood cells have the ability to bind to hemoglobin. Carbon dioxide does. And in some cases it does. That's not how a majority of the carbon dioxide that is in your circulation at any point in time is traveling, but it does have the ability to bind to hemoglobin. Now, carbon dioxide does not bind to the iron ion. What it binds to is the globulin, or the protein portion of the hemoglobin. And when carbon dioxide is bound to the globin part of the hemoglobin, it is referred to as carbaminohemoglobin. Now, in circulation, only about 24% of the volume of carbon dioxide that you have circulating in your blood at any point in time is going to be bound to hemoglobin, forming this carbaminohemoglobin. An overwhelming majority, 76% of the carbon dioxide is going to travel dissolved in the plasma. So when it's dissolved in the plasma, carbon dioxide is going to travel mostly as a bicarbonate ion. So I know in my Bio 103 class, we did an experiment where you took a straw and you blew into water, and as you exhaled into the water that had bromothal blue in it, the color of the water changed from blue to yellow, and that was an indication that the pH in the water was dropping, because when carbon dioxide dissolves in water, it actually makes the liquid slightly acidic. Now you would say, well, I thought you said that the blood is at a constant pH of 7.3 to 7.45, and that's definitely not acidic. And if 76% of the carbon dioxide in your blood is dissolved in the plasma, wouldn't that make our blood a little bit acidic. Well, it doesn't, because there is an enzyme in red blood cells that is going to convert the carbon dioxide and water into first carbonic acid, which is the acidic portion. And then it quickly allows for that carbonic acid to dissociate into bicarbonate ions and hydrogen ions. And the enzyme allows for the conversion of bicarbonate ions and hydrogen ions back to carbonic acid, and that balance keeps shifting one way or the other depending on rising or lowering pH levels in the blood. That being said, if someone has an excessive amount of carbon dioxide in their bloodstream, their blood will become a little acidic. For instance, if someone has suffocated to death, if they have been strangled, if they have been asphyxiated, or have died of a lack of oxygen, shall we say, to their systems, one of the ways that that can be determined is by taking the pH level of the blood, because very quickly your blood, if it does not release that excess carbon dioxide, will become more acidic. So that's one of the ways that you can test in an autopsy to see if that person had a lack of oxygen prior to death, or prior to the heart stopping, shall I say, because if there is a decrease in the pH, or an acidic pH, whatever the word I'm looking to, acid, I'm going to say an acidic, there we go, that's what I was looking for, acidic nature to the blood, that is a sign that it has a high concentration of carbon dioxide, which means there was some kind of issue with breathing prior to death. Again, we're going to talk about that whole entire blood buffering concept much more when we talk about another chapter. But I just want you to know at this point that carbon dioxide, yes, can travel attached to the hemoglobin, not to the heme, not to the iron, but to the globulin protein portion of the hemoglobin. But even though it has the capability of doing so, a majority of it is still going to travel dissolved in the watery plasma. So there is going to be a strict regulation on the level of erythrocytes, or the number of erythrocytes at any point in time that we have in circulation. If you have too few red blood cells in circulation, you're going to have a condition that is generically referred to as anemia. One of the types of anemia is that you just don't have enough red blood cells in circulation to carry adequate oxygen to the body tissues. There are other anemias like iron deficient anemia, where you have enough you don't have enough hemoglobin in the red blood cells. It's a whole class of different types of anemia. Anemia in general just means that you don't have enough oxygen carrying abilities in your blood. But one of those can be due to the fact that you just don't have enough red blood cells in circulation. Now you can go the other way. If you have too many red blood cells in circulation, you have something called polycythemia. And this is an increase in the number of red blood cells in circulation. And this also can be an issue, because as I talked about when I was talking about blood viscosity, the major player for increase in blood viscosity is those red blood cells. And the more viscous the blood is, the more chance you have of having a very high blood pressure, because it's going to take more force to move that viscous, thick blood through the arteries and veins. And you also have a increased chance of that viscous blood just becoming clotted and stuck in smaller vessels. There's an article in your reading talking about blood doping, where athletes will you know, they can, you know, take, you know, have their blood taken, you know, before, you know, a sporting event. And then they can actually spin the blood and remove the plasma and reinsert their own red blood cells so that their red blood cell count goes up. Now, this is a very tricky way of doing it, because if you would test, you really wouldn't be able to tell that they had done anything fishy, because they're giving themselves back their own red blood cell. But this is a problem because, yes, it's going to increase their oxygen carrying ability of their blood, but which, you know, more oxygen to your muscles, the more you're going to be able to provide energy to your muscles. But the problem is that the blood gets very thick. And you add that in with someone sweating a lot when they're doing some sort of athletic activity, that's going to make it even more thick. And you run into the issue of them having an increase in blood pressure and just dying, you know, during the event due to stroke or pulmonary embolism. Another way that people do this to elevate their red blood cell count is to give themselves erythropoietin injections. Now, of course, you can test for erythropoietin. Now, your body does produce erythropoietin, but it's only supposed to produce it at a certain rate. You know, it's kind of fishy. And then there's artificial, like erythropoietin and things like that. But there's a whole article in there. I think there's a video too to watch about Lance Armstrong, because, you know, it's one of the things that he did. So not a really great idea to do. I mentioned the term hypoxia. Hypoxia is low blood oxygen level. This could be due to too few red blood cells or low hemoglobin level. A lot of people now who have the coronavirus are suffering from hypoxia because they're just not getting enough oxygen in as they're breathing. Again, if your body's response to hypoxia, one of them is to release more erythropoietin to try to make up for that deficit in the number of red blood cells. Life cycle of erythrocytes. Erythrocytes are not, like, you're not dealing with the same erythrocytes that you produced when you were, you know, a kid. This is a constant renewal. You're constantly producing new red blood cells. There are several trace elements that your body needs, essential elements that your body needs in order to produce healthy red blood cells. Of course, iron, you're going to need that iron because it's the iron ions you're going to need in order to have that, to bind the oxygen in your hemoglobin structure. Copper, you need copper because some coppers are components of plasma proteins that are going to be used in the production of red blood cells. Zinc facilitates the synthesis of heme. Of course, you need the heme because without the heme, you're not going to have the iron that's going to be there attached to the polypeptides. And then also assorted B vitamins. B vitamins in general facilitate the proper DNA synthesis. A big B vitamin is the folic acid, and folic acid is always a big requirement of pregnant women to take in their prenatal vitamins because it helps for proper DNA synthesis. And of course, if you're making any human and you're making a whole lot of cells all the time, that, you know, you want to make sure that that process is going properly. Erythrocytes live about 120 days in circulation. So what is that, like about 4 months? Eventually, the erythrocytes are going to become worn out, mostly because they do not have organelles or nucleus. So no nucleus, no organelles means no new proteins, no new construction inside the cells going on. So if something starts to break down, you know, it's broken down. You're not going to be able to replace it. Unlike our other cells that have organelles, if, you know, we need a new protein, you know, shoot it off, you know, make some mRNA. Send it out in the cytoplasm, you know, use the ribosomes, Golgi bodies, you know, endoplasmic reticulum, make that new lipid, make that new protein. Erythrocytes don't have this. They don't have the nucleus. You can't make new mRNA. They can't translate proteins. So eventually, especially because they have to fold themselves over and they have that spectrum, eventually, you know, like that pop-up tent, you fold it and you pop it up, fold it up and pop it up several times, you know, eventually those little things are going to become brittle and they're going to break. It's exactly what happens with erythrocytes. They become worn out. They're not capable of folding themselves in half or you know, squeezing through the small places that they could before. They tend to get stuck in the spleen. The spleen has a really small vessels and areas that they need to get through, and they tend to get stuck there. And that's a good thing because there's a lot of macrophages there, cells that devour worn out cells, and they get taken out of circulation. But no problem because we're constantly replacing them. So our body does a really excellent job at recycling all of the components of that are in the red blood cell. So what do we have in the red blood cell? We have mostly hemoglobin. What's hemoglobin made up of? Well, the heme portion, the iron that's attached to the heme, and the globin portion, or the protein portion that makes up those 4 polypeptide chains. So the globin portion is going to be broken down. So here we have our red blood cells. It's going to be taken over, eaten by a macrophage. The particular macrophage is called a Kupfner cell. You don't need to know that. So it breaks it down and releases the hemoglobin. The globin portion is going to be broken down into its individual amino acids. The heme portion that has the iron on it, it's going to be separated. The heme is going to be separated from the iron. The iron goes back to the liver, which eventually it's going to be going back to the bone marrow so they can be reused in the production of new red blood cells. The heme, there's a longer process that goes on here, but this is going to be very interesting, and I promise. Okay, so the heme is a pigment. So the heme goes and is converted to a substance called bilirubin. And bilirubin then is going to be converted to bile. And you may have heard of bile. Bile is produced in the liver and it's stored in the gallbladder, and it's used for the digestion of fats. The heme is reused to produce bile, and the bile is used to digest fats. But the bile helps digest fats, but it does pass through the digestive system, and the bile is then converted into another pigment called stericobulin, I believe it is, and eventually it's converted into a pigment, and it is, we kind of like poop it out in our feces. So here is the take-home question here. So why is your poop brown? Well, your poop's brown because it is colored by the heme that has been broken down and converted into new pigments. So, so now you can pass that along, preferably not during dinner or any kind of meal to other people. And I don't know if they'll be impressed or grossed out or just weirded out, but that's why your poop is brown. All right, leukocytes. I'm going to start talking about leukocytes. I don't know if we're going to get them all through this in lecture, but leukocytes. So again, L-E-U-K means white. Cyte means cell. So leukocyte means white blood cells. So the main function of the leukocytes is going to be to protect the body against invading microorganisms. They also are going to protect us from exterior. This would be the external threat of bacteria, viruses, fungi, even parasitic worms. There is a white blood cell that is specific to taking out parasitic worms, which is scary because how often do we get infected, or did we get infected with parasitic worms that an entire cell line is committed to this? Leukocytes also can destroy cells that have any kind of mutated DNA. As I mentioned before, there are white blood cells that are dedicated to just fighting off these mutated DNA cells. Mostly, these are the type of cells that reproduce quickly and are the cause of some cancers. White blood cells also are going to be there to just clean up general debris. They're the janitors, let's say, of the blood. A normal white blood cell count is about 5,000 to 10,000 white blood cells per microliter. There are 1,000 microliters in a milliliter, I believe. So a microliter is about a drop. So if you were to pick up an eyedropper and one drop of water, that is around a microliter. So 5,000 to 10,000 per microliter. If you have a elevated white blood cell count, obviously there is some sort of infection or something that's going incorrectly, that production of the leukocytes have been upped. And based on the exact type of cell number, because each of the 5 are going to take care of something specific, you can actually kind of narrow down to what is going on. If you have a level that is too low, well then obviously there's also some sort of issue going on. So leukocytes are really the only formed element that in circulation are complete cells, because they're the only formed element that are going to circulate with a nucleus and all of the organelles. Red blood cells start off circulation as reticulocytes, no nucleus, some organelles, but when they're mature, they have no organelles, no nucleus. Platelets, there's no nucleus, there was no organelles to start off with. It's really kind of just the cytoplasmic extensions that have broken off of a larger cell. But our leukocytes here are real players. They're going to have nucleus and organelles. Because of this, they're going to be much larger than the erythrocytes. However, they have a much shorter lifespan than erythrocytes. Even though they have these nucleus and this organelle so they can repair themselves, they last from about hours to minutes. And the reason why they have a shorter lifespan mostly has to do with the fact that they die in the process of doing their jobs. Their jobs are to kill invading, pathogens, or to take out and destroy debris. And a lot of times they sacrifice, well, most of the times they sacrifice themselves to do so. So they do not have a very long lifespan. A lot of the jobs that leukocytes do, they do because they are able to leave circulation. They're able to leave the capillaries. And they do so through a process called diapedesis. So here we have an illustration of that. We'll learn about these different pores and fenestrations and openings that we find in capillaries. And these openings are large enough for these white blood cells to actually squeeze through these openings and move into the surrounding tissues. And the surrounding tissues are going to be where they're going to find these foreign particles or abnormal cells that they need to get rid of. So in some cases, and I believe your reading really focuses on the word emigration. Yes, that is correct. I usually have heard diapedesis more than emigration, but just be aware that they use that term interchangeable. Now, once in the tissues, the leukocytes are going to move around the tissues, kind of looking for the troublemakers through a process called amoeboid movement. And the reason why it's called amoeboid movement is because they move like amoebas. And if you remember, we talked about amoeboid movement in 103. The amoeboid movement is kind of when the cells will like extend their cytoplasm towards one way, and then they kind of like suck the other cytoplasm towards that way. So they kind of like, please do not, I just realized I was doing that. I so hope that I don't like show up on like a meme like doing this, but anyway. So they kind of like squish and kind of like roll their way around in this amoeboid movement. They aren't moving just haphazardly though. They move towards what are referred to as chemotactic chemicals, or chemical signals that other cells have released. So when a white blood cell is, out, checking things out, if they come across something that needs to be destroyed, they will start the process of destroying that pathogen, but they will also release chemicals into the tissues that will signal the alarm for more leukocytes to come that way. So these chemotactic chemicals are kind of like warning signals or tracers for other leukocytes to follow, so they know that they're needed in a specific area. And the process of movement towards these chemical signals is referred to as positive chemotaxis. Positive meaning towards, because if you're going towards something, it's positive. If you're going away from it, it's negative. So positive means towards. Chemo refers to, it's a chemical cue, and taxis means movement. Like you take a taxi somewhere because it's movement towards. So movement, chemical, towards chemical movement. So movement towards a chemical cue is positive chemotaxis. Now, there are 5 different types of leukocytes, and we can take those 5 different types and we can put them into 2 general classifications. The first classification is granulocytes, and a granulocyte, this category is called that because there are a lot of granules inside of the cytoplasm. These granules are basically just vesicles that are filled with different chemicals. And the granulocytes are made up of the Phil family. So we have neutrophil, eosinophil, and basophil. The reason why they're called neutro, eosinophil, and basophil has to do with how they stain on a microscope slide. So the eosinophils react with an acid-base stain. The basophils react with a basic stain, and the neutrophils react with a neutral stain. So that's how they got their names. And what are we staining for? Well, they're staining for these granules that are actually located inside the cytoplasm. The other classification of leukocytes are the agranulocytes. So A means without. So agranulocytes are going to contain fewer and less obvious granules in the cytoplasm. And when you see pictures of those, hopefully you'll be able to see the difference. And these include the monocytes and the lymphocytes. So not all of the leukocytes are present in equal numbers in circulation. So we have ones that are very, very prevalent, and we have ones that are kind of rare. So the way that you can remember the most prevalent to the most rare is with the little saying, never let monkeys eat bananas. So if you remember never let monkeys eat bananas, you will remember NLMEB. And that is from the most prevalent to the least prevalent leukocytes. So we have neutrophils for never, so that's the most prevalent. Then lymphocytes for the let, monocytes for the monkey, eosinophils for the eat, and the basophils for bananas. So if you were to take a blood sample, you would expect to find mostly neutrophils, and the least amount would be basophils. And you would expect neutrophils to be most numerous, then lymphocytes, monocytes, eosinophils, and then basophils. So never let monkeys eat bananas. All right, so let's talk about these guys individually. So we're talking about the granulocytes first. So granulocytes, the first one we're talking about is neutrophils. Because they're the most common, they make up about 50 to 70% of any of the leukocytes that are in circulation at any point of time. The way that you can tell, and again, I know that you're probably going to look at these and be like, Lady, you are crazy. I cannot tell the difference between any of these. But I am going to tell you what I look for, and what I look for, of course, it's easier for me to notice it because I already know this stuff and you guys are just brand new to this, but I'm trying to give you any kind of help that I can possibly give you, is that the neutrophils, when you look at them, all right, so this is the nucleus here, this dark thing right here. This is all the nucleus. So the nucleus is referred to as lobed. So normally when you see a cell, you expect it just to have this round portion in it, and that's the nucleus. But the nucleus of the Phil family, all of them are highly lobed. So it looks like it's, all over the place. And you have to think of, you're looking at this in a flat dimension. So if I have a nucleus that looks like, say it's curved like this. Well, if you look at it from the top, well, it would look like this. If you look at it from the side, it would look like this. If you look at it from this side, it looks like this. But if you look at it from the side, well, now it looks curved. So think about it this way here. So we're looking at this three-dimensional shape from a flat view. So it gets this kind of wonky, distorted look. But this dark purple portion, the whole thing, that there's a nucleus. Now, I said the granulocytes, one of the things that they have is these small granules that are located in the cytoplasm. So these granules here, they look a little bit dotted to me. Again, they may not look that way to you, and I understand. But you can agree with me that this cytoplasm in general is a light purple, especially when you compare it to the color that's in the nucleus. All right, so when you're looking at a neutrophil, you're going to have that crazy looking nucleus, and the cytoplasm may have a dotted appearance, but at least we know that it is a light purple color. Now, neutrophils are rapid responders, and that's why we have so many of them. They are quick to act, and they are phagocytes, or they are cells that eat other cells. So they're going to rush to the scene, and they don't ask questions. They're just going to, eat whatever kind of debris or foreign particle that they come in contact with. The granules that are inside of the cytoplasm that give the cytoplasm this kind of light purpley dotty appearance contain two different chemicals. The first one is a lysozyme. A lysozyme is a type of enzyme that is going to be capable of breaking down bacterial walls. So along comes the neutrophil, it sees a bacteria, it engulfs it, it brings it inside, and then it's going to release those lysozymes from the granules and basically dissolve away the bacteria after it's engulfed it. Another type of chemical or molecule that it's going to contain within its granules is called defensins. And defensins are a protein that is going to be able to also bind and puncture through bacterial plasma membranes or bacterial coverings. So basically, our neutrophil comes along, it sees, some bacteria, my phone, it grabs onto that bacteria, and once the bacteria is inside of the cell, because the cell has engulfed it, it's going to release lysosomes to break down the bacterial cell wall. Then once the cell wall is gone, it releases defensins and breaks down that plasma membrane so that it basically destroys the cell. Now, if you've ever gone to a doctor and they say, oh, we're going to do a white blood cell count because you don't feel good, we're going to find out whether or not you have a virus or you have a bacterial infection. Well, how do they know that? By looking at my blood. Well, if you have a bacterial infection, you are going to have a very large number, even higher than you normally have, of neutrophils in circulation. Because when you get an infection by a bacteria, neutrophil production is going to be ramped up so that there's plenty there to attack, digest, and get rid of the bacteria. So if they take your blood, they put it on a microscope side, they start scanning it, and they're like, wow, these neutrophil numbers are outrageous. They're like everywhere. Chances are you probably have a bacterial infection. They're going to give you some sort of antibiotic and hopefully send you on your way, and you'll be good to go. All right, so when you think of neutrophils, think of most numerous, think of light purple, think of killing bacteria. Our next granulocyte is the eosinophil. Now, again, we have this crazy looking nucleus. It's not just one round circle. It's all, it's kind of connected with each other. Again, why? Because this is a lobed nucleus. It's a three-dimensional structure that we're just looking at from one section. Now, these guys, because they're granulocytes, also have granules in the cytoplasm, but their granules are going to stain red. So this is all the cytoplasm, and again, it has this dotty appearance. But as you can tell, this cytoplasm is red compared to this cytoplasm, which is light purple. Hopefully you can tell the difference between that. So this is light purple, hopefully not colorblind. If you are screwed, probably. I'm so sorry if you are. So this is light purple, the neutrophil, while the eosinophil is going to be a red color. Now, the granules inside of a eosinophil is going to contain antihistamines. So you have heard of antihistamines. It's what you take when you have an allergic reaction. So antihistamines counteract the activity of histamines, hence antihistamines. And histamines are an inflammatory chemical. The granules also are going to contain cytotoxic proteins or cell toxic proteins. So what do these granulocytes do? So they're going to be very important. These are the ones that are important in defense against parasitic worms. So essentially what these guys do is if you have some sort of parasitic worm, they work in groups and they will surround the worm, and then they kind of like split themselves open and they release their cytotoxic chemicals, and the cytotoxic chemicals will then start attacking the worm and kind of digest it from the outside in, which is pretty horrific, but I really don't want any kind of parasitic worm in me, so I'm cool with that. The eosinophils also can work to, they have a phagocytic kind of a function where they can attach to and engulf antigen antibody complexes. And take a note on that and just kind of put a pin in it, because we're going to talk about antigen and antibody complexes later on in this lesson, not in this lecture. So what if you have a high count of eosinophils? So if you have a high count of eosinophils, a couple things can be happening. The first thing is you may be suffering from an allergic reaction. Why would you have a lot of eosinophils? Well, I said back here that the granules contain antihistamines. So if you're having an allergic reaction, normally what happens is your body produces a lot of histamine. It causes red inflammation in that particular area. So if you're having an allergic reaction, these eosinophils can go there, release their antihistamines to try to calm down the situation. Basically, this is your body's natural Benadryl. It's going to respond to that allergic reaction, release its antihistamines, and kind of, calm the situation. Also, you could have a parasitic worm infection. Let's hope that's not it. And also, there are cases where there are certain autoimmune diseases where your body attacks itself that will cause a rise in eosinophils. The last granulocyte are the basophils. All right, so here we have, you can kind of see it, the lobe nucleus, like here in the background, but it's obscured because the granules in the basophil are very dark blue, dark purple. All right, so let's review. Let's go back to this guy. So this is our neutrophil, lobed nucleus, light pale purple cytoplasm granules. Eosinophils, lobed nucleus, red granules in the cytoplasm, and then basophils. Basophils, lobed nucleus, dark purple, blue, whatever you want to call them, granules in the cytoplasm. Sorry. Sorry, I inhaled real too quickly, sorry. So, basophils are the least common leukocytes. If you remember, it's never let, sorry, I need to get another drink. Never let monkeys eat bananas. So this is, basophils are about less than 1% of the leukocytes that are in circulation. So basophils are going to work to intensify inflammatory responses. So kind of the opposite of what eosinophils do. Their granules are going to contain histamines, which are going to contribute to inflammation. And they also contain heparin. Now, heparin is going to be what we refer to as an anticoagulant. It opposes blood clotting. And heparin is used a lot in medicine. Well, heparin is used in medicine when you're giving IV It prevents the blood from getting clotted in the needle itself. Heparin's also used therapeutically when people have blood clots or it's at risk for strokes. But our body also naturally produces heparin, and one place that it is going to be stored is going to be in these basophils. So if you have a high count of basophils, you are probably suffering from some sort of allergic reaction. The allergic reaction is caused by this massive release of histamine from the basophils. It could also be a parasitic infection and also hypothyroidism, or too low functioning of the thyroid gland. I believe it's also basophils that are elevated in individuals who are having an active asthma attack. All right, so that has been an hour. So we're gonna stop here. When we pick up next time, we'll talk about the agranulocytes.